Hi, I'm Joanne Jezequel, a biology teacher for high school and college. Let's talk about cells, the smallest living units of an organism. You need to know that all cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. You also need to know that there are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they are customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Now, some organisms, such as plants that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic, hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.
What is the structure and function of a cell? Well, there are three common parts exist in any cell, like cell membrane, cytoplasm, and nucleus or DNA. Now, what is cell membrane? It is the selective permeable membrane which allows certain materials to pass through. For example, it allows essential ingredients to enter to the cell like water and oxygen. Secondly, cell membrane remove toxic substances out of the cell like carbon dioxide and waste products. Thus, cell membrane allow good products to enter to the cell and remove waste products out of the cell. Now, what is cytoplasm? Cytoplasm is the thick jelly-like structure and it provides shape to the cell. It means that different shapes of the cell are due to the different shapes of the cytoplasm. All the cellular functions or cellular reactions take place here in the cytoplasm. For example, protein formation occur in the cytoplasm, respiration occur in the cytoplasm, mitosis or meiosis occur in the cytoplasm. Secondly, all important organelles are present in the cytoplasm like mitochondria, ribosomes, endoplasmic reticulum, Golgi bodies, lysosome, which we will discuss later in this lecture. Now, what is nucleus? Well, nucleus contains DNA. DNA is a booklet of information. This booklet of information is the blueprint of all the functions taking place in the cell. Secondly, nucleus is the control center of a cell or king of the cell. It controls all the activities occurring within the cell. For example, nucleus controls how cell should develop, how cell should survive or grow, how cell should reproduce, etc. Thirdly, nucleus contains nucleolus. It forms ribosomal RNA or ribosomes. The newly made ribosomes are transported out of the cell through the nuclear pores to the cytoplasm where they can do their job. Now, what are ribosomes? Well, ribosomes are floating organelles in the cytoplasm. They make proteins needed for the cell. For example, ribosomes makes protein for the growth of the cell. Ribosomes make proteins for the repairing of the cell. To learn about the ribosomes and protein formation, we must understand endoplasmic reticulum. Endoplasmic reticulum are of two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. The main difference between them is that rough endoplasmic reticulum contains ribosomes and smooth endoplasmic reticulum does not contain any ribosomes. It means that smooth endoplasmic reticulum does not produce any protein because it has no ribosomes, but it produces lipids or cholesterols, etc. Secondly, it also does detoxification. It means that breaking down of the toxic substances within the cell. Now, coming to the rough endoplasmic reticulum. We know that it contains ribosomes and ribosomes form protein. Then what is the function of rough endoplasmic reticulum? Well, it helps the ribosomes to produce proteins. Secondly, rough endoplasmic reticulum pick or enclose the proteins and vesicles. Now, what are vesicles? Vesicles are the trucks within the cell. Vesicles are the truck inside the cell that transport substances within the cell from one part to another. The protein found by the ribosomes and packed by the rough endoplasmic reticulum is transported by the vesicles to the Golgi body, also known as a Golgi apparatus. Golgi is the name of the scientist who discovered it. Remember that Golgi body is the port of the cell, where things are collected, packed and exported. Golgi body receives proteins from rough endoplasmic reticulum. It modifies the protein by giving them proper shape. For example, it will add lipids or carbs to the protein and change the shape of the proteins because shape of the proteins play a pivotal role in its function. After this, Golgi body will pick the protein in vesicles. Finally, vesicles of the proteins are delivered to the targeted destination both within or outside the cell. For example, Golgi body will send proteins to the damaged part of the cell for repairing by the help of vesicles. Until now, we learned that ribosomes are formed by the nucleolus and the nucleus. Ribosomes synthesize proteins. A rough endoplasmic reticulum pack the protein in the vesicles and transport them to the Golgi body. Golgi body provides specific shape and prepare the protein for a specific task. Finally, Golgi body sends it to the targeted destination to perform its function. Now, what is mitochondria? Well, mitochondria is the power station of the cell. For instance, in our daily life, power station provides electricity or power to the city. Similarly, mitochondria provides energy to each and every part of the cell. Mitochondria also contains its own DNA, different from the DNA of the nucleus. In mitochondria, cellular respiration takes place. For example, glucose will combine with oxygen to release energy in the form of ATP, and carbon dioxide gas is produced as a waste product. Energy produced in the form of ATP by the mitochondria is then transported to each and every part of the cell. Now, what is lysosome? Well, it is digestive part of the cell. It contains digestive enzymes. Here, food is broken down. For example, like carbohydrates are converted to glucose, proteins are converted to amino acids. Finally, what is vacuole? Well, vacuoles are called storehouse of the cell because vacuoles store water, food, mineral salts, nutrients, waste products, etc. The size of vacuoles may vary in different organisms. For example, in animal cell, there are many smaller vacuoles, while in plant cell, there is only one and large vacuole. Now, what about animal and plant cell? Well, to learn about them, watch our next lecture.
Or in other words, what is the building block of an organism's body? The cell, that's absolutely right. Cells make up the bodies of all living organisms. How many cells do you think an organism needs to live? 1, 2, hundreds? Well, now that depends on the organism we are talking about. An organism like amoeba is made up of only a single cell. Yes, you heard it right. Just one single cell constitutes the body of an amoeba. This cell carries out all the functions of this organism's body, be it respiration, digestion, locomotion, and excretion. What about a cat or a dog? Do they have a single cell? Of course they don't. They have many cells which make up their entire body. There are groups of cells which carry out different functions.
Development is a process of progressive and continuous change that generates a complex multicellular organism. It starts off with the gametes, where the sperm fertilizes the egg through a species-specific process. This creates the zygote, a fertilized egg with a diploid chromosomal complement generated by the fusion of the haploid male and female pronuclei. After fertilization, the zygote goes through several developmental phases to create an embryo. The embryo consists of a developing organism prior to birth. In mammals, the embryo refers to the early stages of development until the end of the organ-building phase, known as organogenesis. After this, the developing mammal is a fetus until birth. At birth, it becomes a neonate. And it doesn't stop there. Development progresses to the maturation of the adult form and continues into senescence. A major achievement in descriptive embryology was the idea of a generalizable life cycle, where each animal species passes through similar stages of development. The first is fertilization, fusion of the mature sex cells, followed by fusion of haploid gamete nuclei to restore the full complement of chromosomes of that species. Next is the cleavage stage, a series of rapid mitotic cell divisions that divide the embryo without increasing mass. During cleavage, the cell cycle consists primarily of the S and M phases, otherwise known as the DNA synthesis and mitosis phase. The gap phases, G1 and G2, are essentially skipped. As a result, there is no increase in mass with subsequent divisions. This allows the cytoplasm to partition into many smaller cells called blastomeres. At the 16 to 64 cell stage in a vertebrate embryo, the blastomeres develop into a solid, berry-shaped ball called a morula. After further cleavage divisions, it comes to a point where cleavage comes to an end as the blastomeres form an early stage embryo consisting of a hollow sphere of cells called a blastula. If you took a cross-section at the blastula stage, you would see that the blastula surrounds an inner fluid-filled cavity called the blastocele. The next stage in embryonic development is gastrulation, a dramatic reorganization of the hollow blastula into a multi-layered embryo called a gastrula. Here's an early gastrula, where the cells move during gastrulation to take up new positions and acquire new neighbors. At the start of gastrulation, a group of cells will buckle into the blastocele to form a shallow depression. Continued invagination forms the archenteron, the rudimentary alimentary cavity of an embryo. The open end of the archenteron and the first opening formed in the embryo is the blastopore. The blastopore is important because this will either become the mouth or anus, depending on the type of triple blastic organism we are talking about. In protostomes, the mouth forms from the blastopore. In deuteriostomes, the anus forms from the blastopore. As cells take up new positions, the archenteron will extend through the embryo to reach the surface at the other end to a complete formation of the primitive gut. The embryo is said to be a gastrula when it contains the 3 germ layers that will interact to generate the organs of the body. During the differentiation process, we concern ourselves with the ectoderm, mesoderm, and endoderm. These are the germ layers. When a zygote starts dividing and undergoes morphogenesis, the movement of cells, there are various layers formed that have a fate or derivative set of cells or tissues that it will become, due to the nature of where it is found in the embryo. We use the universal colors blue, red, and yellow to represent the 3 germ layers. Blue is for ectoderm. The ectoderm are the cells that remain on the outside surface of the embryo following gastrulation. These cells will form the nervous system and the skin. Red represents mesoderm, the middle layer that gives rise to muscles, bone, connective tissue, reproductive organs, kidneys, blood, and most of the cardiovascular tissue. It is important to know that lower phyla like Porifera and Cnidaria contain 2 primary layers that give rise to all the tissues and organs of the animals, making them diploblastic, as they lack a true mesoderm. The third layer is the endoderm, represented by yellow. This is the innermost germ layer that forms the epithelial lining of the respiratory tract, gastrointestinal tract, and the accessory organs of the digestive system like the liver and pancreas. For the DAT, it is very high yield to have a basic understanding of what the derivative of each germ layer are. The last major stage of embryonic development is organogenesis, where cells in the regions of the embryonic germ layers interact with one another through chemical signaling and rearrange themselves to develop into tissues and organs. An example of organogenesis is the process of neurulation. This begins as cells from the mesoderm form the notochord, a rod that extends along the dorsal side of chordate embryo. Signaling molecules secreted by these mesodermal cells cause the ectoderm above the notochord to become the neural plate. This is an inductive interaction as a group of cells influences the development of another group of cells through a close-range interaction. After the neural plate forms, cells begin to change shape, which curves the structure inward. The neural plate rolls itself into a neural tube that will become the brain and spinal cord. The notochord will degenerate before birth, although parts of it will persist as the inner portions of the intervertebral discs.